today. Please join me in welcoming Dr. Aprajita Singh. Dr. Singh completed her Bachelor of Fisheries Science from West Bengal University of Animal and Fishery Sciences in 2014. She went on to complete her Master's in Fish Nutrition and Feed Technology from the Central Institute of Fisheries Education, Mumbai in 2016. Dr. Singh is a recipient of the ICAR Netaji Subhash International Fellowship, under which she recently completed her PhD in Animal Nutrition and Management from the Swedish University of Agricultural Sciences. Dr. Singh has showcased her work at various international conferences across Europe, including the Netherlands, Portugal, and Norway. As an early career research scientist, she boasts an impressive record with 4 international research publications. Additionally, Dr. Aprajita is an active member of both the World Aquaculture Society and the European Aquaculture Society. She also contributes as a reviewer of Aquaculture International. So please join me in extending a warm welcome to Dr. Aprajita. The floor is now yours, and we look forward to your enlightening talk. Thank you.
And thanks, Deepak, for the warm welcome. Now I will share my presentation.
Yeah, sure.
Hope this is visible to everyone.
Yes, it is. Thank you.
Okay. First of all, thanks for inviting me on this platform of AFS Talks. Today I'm going to talk about my PhD work, which was on alternative feed sources, effects on gut microbiota, immunity, and health of rainbow trout. So my presentation will be consist of different sections, starting from the introduction, aim of my research work, experimental design, methodology, results, conclusion, and acknowledgement. But I would like to inform that I will not be including everything which I have done during my PhD period, but I will include some of my important findings in the results, and I would like to discuss about them now. But if you want to know more about what I have been doing and what I have done in my PhD work, you can always go to the SLE website where my PhD thesis has been published. And also I have attached a link at the end of my presentation, so you can also go through that. So traditionally, the aquaculture feeds were consist of fish meals, which are not very sustainable. So with the time, they has been replaced with various plant-based ingredients such as soya and rapeseed meal and other kind of mustard seeds, seed cakes. But these ingredients are also not very sustainable in long runs because they need for edible lands, water, and also they have huge environmental load. At the same time, they compete with the human as a feed sources. Apart from this, they have anti-nutritional ingredients, which hampers the fish health in long term. So there is a need for different kind of or alternative feed substances, or we can say a new generation feed resources, which can be obtained from organic or organic waste or the forest waste or industrial byproducts. So these feed ingredients should be in low environmental footprints. They can be like cost effective, and also at the same time they contribute to the 3 principles of R, that is reduce, recycle, and reuse, that's contributing to the biocircular economy. The special quality of these ingredients are they can observe or they can take the nutrients from the substrates on which they are growing. So when these ingredients are ingested into the body, then they get assimilated and microbial transformations, which leads to the good health, gut health, immunity, and also improves the overall well-being of the fish. Today, I will talk about 3 of these kind of ingredients, which I have been using in my research work. The first 2 are the microbial biomasses, Urospora intermedia and Yarevia lipolytica. And these ingredients has been considered as safe to be consumed by the humans and the animals. These molecular biomasses have high protein content, or they have a good nutritional profile. And at the same time, they have bioactive components such as glucan, chitin, chitosan, and mannan. The third ingredient which I will be using is a cellular oligosaccharides. It is a 3 to 10 short chain beta-glucopyranose units. They are produced by a controlled enzymatic hydrolysis of lignocellulose. And it has been seen that they had improved the host digestion intestinal ecology. But this is the study using these ingredients for the very first time. So we do not know like how these ingredients behave when they are ingested in the gut, and how they're going to impact the gut host interactions, or how they're going to impact the immunity or the gut barrier function of the fish. As we know that the gut harbors millions of bacteria, and these bacteria help in providing the nutrition by producing different kind of digestive enzymes, or maybe they can help in the development and the growth of the gut associated lymphoid tissues, which plays an important role in improving the immunity or immunomodulation of the gut. But also, this hemostasis of host and the gut microbiota is affected by the abiotic and biotic factors. When these factors are adverse, or we can say like when there is an invasion of the pathogen, then this hemostasis breaks, and there is a situation that leads to a disease or which can be harmful for the fish. It has also been known that beta-glucan is able to contribute immunomodulatory effects. As we know that the beta-glucan acts as a microbial associated molecular pathways, and they trigger several kinds of molecular pathways such as toll-like receptors, C-type lectins, and complement systems. At the end, they are able to improve the adaptive and innate immunity of the fishes. So the overall aim of my study is that I'm willing to find the potential of this kind of microbial and cellular oligosaccharides on the fish performance, whether with respect to the gut microbiota composition, their effects on mucosal immunity, or any kind of stress parameters in rainbow trout. The experimental design consists of four papers, in which paper 1 and paper 2 uses Neurospora intermedia as a test ingredient. The study is conducted for 30 days, and the different samples were collected at day 0, 10, 20, and 30. And for this, the 3 diets were prepared in the replicates of 5. And the diets were prepared as per the digestibility study in the ratio of 70:30. One of the Neurospora diets were preconditioned to see the effects of the heat on gut microbial composition. Paper 3 uses the Urearovia lapolitica as the test ingredients, and it takes like 45 days for the feeding trial. Here the yeast were autolyzed and were included in the ratio of in the percent of 2 and 5 as a whole yeast or autolyzed yeast. The paper 4 uses the cellular oligosaccharides, and the studies goes similarly as the 45 days and then the final sampling. But here the inclusion level for COS follows as 0.1%, 0.5%, and 1.5%, whereas FOS used as a positive control. So here the feeding trials and the sampling was done at the fish technology lab at SLU. And the fishes were first stored in the holding tank prior to during the acclimatization period, and then they were shifted to the experimental tanks for acclimatization for some day. And then the experimental tanks were connected with the automatic feeders and the fishes and feed collecting chambers. The feeding was done twice a day, and this setup has automatic controlled water flow, and also the water flow was maintained at the temperature of around 11 degrees plus and minus, and the light were maintained for 12-hour day and night. And at the end of the feeding trial, the samples were collected from as a blood and the tissue samples. After collecting the samples, they were analyzed for gut microbiota analysis using 16S RNA sequencing for the V3, V4 region, and for gene expression, it was done through qPCR technology. So now discussing about the results, I will go by one for every chapters. And for the paper 1 and 2, I will be looking into the digestibility data and the gut microbiota and its effect on gut microbiota at different time intervals. So this table shows the feed formulation and the proximate composition of the diets. And here you can see that the 30% of the reference diet has been replaced with the test diets, which is as per the requirement for the digestibility study. So, and the digestibility of Neurospora intermedia was studied at day 30, and which shows that the Neurospora intermedia is highly digestible in terms of diameter digestibility, crude protein, crude fiber, and gross energy digestibility. But it has also been seen that the heat does not have any effect on improving the digestibility, because it was one of our assumptions, like the heat will improve the digestibility as it breaks down the starch, but we could not find anything. So, but here the diet Neurospora intermedia, which has not been treated with the heat, is showing good results as compared with the reference diet. Beta diversity of microbial composition was studied with proximate coordinate analysis, and it shows that the diets at the day 0, so the diet at the day 0 have similar kind of gut microbial composition, whereas the diet when at the day 10 and day 20, there are differences in the gut microbial composition. Perhaps again at the day 30, all the microbial biomass look similar for each diet. So this can be explained like in the very beginning, in the 0 day, fishes were acclimatized and they were fed with the commercial diet. So it shows it has all kind of similar kind of composition. But after 0 day, the actual feeding trial started and the Neurospora intermediate diets were given to the fish. So it took some time for the fish to acclimatize with the changing environment of the diet. And again, at the day 30, they became the similar kind of microbial composition. This suggests that the gut microbiota composition is affected by the diet and the time of the feeding period. So according to these results, the feeding trial should be done for 30 days or more than 30 days to see the pattern of gut microbial composition due to diets. So relative abundance of different gut families were observed, or different microbial community were studied, and it shows that the interaction plots for the Peptostreptococcus has a decreasing trend for the abundance, whereas the Lactococcus has the increasing trend for the abundance. So you can see there is a decrease in the Peptostreptococcus from day 0 to day 30, whereas it increases from day 0 to day 30 for Lactococcus lactis. So originally, the Peptostreptococcus has an affinity for the animal protein sources or substrates. They like growing on those substrates. So when there is a change in the substrate from 0 day, which was a commercial diet, the plant-based diet, that is or fungus-based diet, that is a Neurospora intermedia, so their abundance decreases. Whereas it has been seen that the Lactococcus lactis or the lactic acid bacteria strains were favored when they were fed with Neurospora intermedia. So for the short conclusion for this project it can be stated that the Neurospora intermedia is highly digestible, and the replacement study as a protein source is suggested. And at the same time, the diet and time have effect in shaping the gut microbiota. And there is an increase in abundance of lactic acid bacteria, which has seen to produce the short chain fatty acids, which has a different kind of functions in regulating gut homeostasis. In paper 3, the potentiality of Yarovia lipolytica has been studied. So here, the inclusion level of 2 and 5% has been maintained by replacing some part of soy protein concentrate and the methyl cellulose. So coming to the microbial composition study for this particular yeast, there was not a lot of influence on the modulation of the gut microbiota, as there was no significant difference in the alpha and beta diversity. So it can be stated that it has a limited effect on the gut microbial composition. But when there was a log2 fold chain study done, which is a study to see like which microbial families are synergistic to the diet, then it has been observed that disulfovibrio were more synergistic to the diets with whole yeast, whereas the Spingobacteriaceae were more synergistic with the diets which have higher inclusion level of yeast. A total of 11 genes has been studied regarding the immune parameters and the gut integrity, where the beta-glucan and RSP20 were used as a reference genes. The gene expression study reveals that the Yarovia lipolytica is able to influence different kind of pathways, like toll-like receptors, complement pathways, C-type lectin, and IGT and CD4. And the maximum expression were seen in the diets with 5% inclusion level as a whole yeast. So it can be stated that Yarovia lipolytica is able to modulate innate and adaptive immunity. So short conclusion, it can be stated as it has the capacity to enhance the mucosal immunity when it is included as a 5% inclusion level. And also it has been seen that the whole yeast has more antigenicity, or maybe it can be considered as more bioactive than the autolyzed yeast. So the paper 4 studies the effects of cellular-oligosaccharides on the gut microbiota and the oxidative stress and immunity of rainbow trout. To study the beta diversity, the principal component analysis was done to see the correlation of the diets with the microbial composition. And it has been seen that the diets with higher inclusion level of COS, that is 0.5% and 1.5%, has shown inverse relationship with the control diets. That suggests that there is a differences in the microbial composition with the control diet. When relative abundance of the diets with cellular-oligosaccharides was done at a family level, it has seen differences in the Brevinemyceae, Ruminococcaceae, Chitinobacteriaceae, Bacillacea, and Lactobacillacea, which means that the abundance of lactic acid bacteria from the families Bacillacea and Lactobacillacea is there, and it has been seen that the abundance of these bacteria increases with the higher inclusion levels of the COS in the diets. So for the oxidative stress analysis, 3 enzymes were studied: superoxide dismutase, catalase, and glutathione peroxidase. Among them, it has been observed that the superoxide dismutase has higher amount in the serum as compared to the control. So it suggests that the cause has some reactive oxygen, has a capacity to fight against reactive oxygen species, and thus providing more antioxidant abilities. The immune gene expression suggests that there is not much activity with respect to the cause, like the cause is not able to influence it at a high level. There is only significant value for the complement 3, whereas the trend has been seen in TLR2 and TLR and C-taplectin. So at the end, it can be concluded for the cell oligosaccharide inclusion is that the cell oligosaccharides is able to modulate the gut microbial composition, but when included at a higher level, there is no clear immunomodulation effects, and there is a higher antioxidant capacity due to cause. So it can be suggested that a higher inclusion level studies, like it has the maximum inclusion level for the cause is 1.5%. So it has been suggested to include it further more than 1.5% and see how it affects the gut microbial composition, or maybe there could be a sign for immunomodulation as well. So coming to the final conclusion for the whole PhD work, so it can be stated with the Neurospora intermedia can be considered as a protein source and can be experimented with this because it has high digestibility. So the future prospect can be as using it as the protein source in the diets. At the same time, the Yerevia lipolytica does not have much effect on the microbiota, so it can be used as an immunomodulator or additives in the diets. But it should also be mentioned that it has the immunomodulatory effect until the inclusion level of 5%. So it should be good to check how it behaves when included in the higher inclusion levels. The cause can be useful as a functional feed to modulate the gut and the oxidative stress. Apart from this, the success of this kind of ingredients depends on their nature, on the and also on the substrates on which they are growing, because they absorb the nutrients from there. So that is important to consider, and also what inclusion level they have been included, and also the processing method that also play an important role, like the downstream processing method, because it has been seen that the yeast kind of ingredients or fungi kind of ingredients which have a tougher cell wall, they are lower in assimilation and absorption in the gut. So some studies have seen the improvement in the digestibility and absorption due to the processing method. So finally, I would like to thank all my supervising committee for helping with this project works, and my collaborators, which has provided me the guidance and the fundings, and also the funding agencies like FORMAS, Swedish Agency for Ecology Research, Economic and Regional Growth, and Vinova.
I guess that's your last slide.
Yes, this one is my last. Okay. Thanks for your attention. I was not able to turn it.
Yeah. I thought there's more coming. All right. Let me change some settings first. All right. So thanks very much for that excellent talk, and thanks to all who joined today for the talk. So I have put a link in the chat box for you, everyone, to give a feedback, which I will pass to Aparajita once we finish our session. So it's the time for discussion. If any of you have questions, please type in the chat box or use the hand raise feature in the Zoom. So while you think your questions, I will ask a couple of ones that I prepared while you're talking. Now, I'm not an expert, so apologies if these questions are like very simple, or probably isn't that very obvious that or you may be surprised with my questions maybe. Um, so just from a very superficial level, so I guess the research was to reduce the cost of the feed by using alternative sources. Is that right? Yeah.
Yeah, the background for the selection of these kind of ingredients was like that, but
Yeah.
we have not done any economical study.
Yeah. So which is where I was going forward. From an industry perspective, it would be really nice to have some insight into how it's more than reducing the cost for the feed. It's more interesting to know how much it will make a difference in the net of returns. So that means which will include the other indicators such as how fast they grow, in terms of biomass or survival. Uh, was there any other sort of stresses or by replacing this diet? So is there any insight in those things which would really matter for an industry perspective, I guess?
The actual plan for this is like now, like we are testing it as a very small scale, because all these ingredients were produced in the laboratory in different universities here. So they have provided us such kind of ingredients. But thing is, like, until we suggest to an industry, we have to do it or scale it up to some up to a level where we can have like a continuous supply of the ingredients, like continuous supply or continuous production of the ingredients. So that has not been reached yet, because these are produced in very small amounts in some of the labs. So they do not have the infrastructure to make it on a large scale and
Yeah.
to do a industry kind of. So that is also a constraint. But if an industry is interested to invest in this, then could be really nice to scale it up. And also at the same time, I will say, like, these ingredients are like growing on the waste or organic waste. Like for the first one, like Neurospora intermedia, it has been produced from the byproduct of the industry waste. So they are just using the waste from the industry. They are not using something like new kind of ingredient or the land or something kind of facilities. So they are growing them on the culture media, and they are then fermenting and extracting it, and then we are doing the studies.
Yeah.
Similarly, some from the sewage, they also getting fermented in the small fermenters, not from not on the very large scale. The studies, that's why the studies are in the very small scales.
Yeah, I know it's a long way ahead to reach what
Yeah.
we were talking. My question was more on your gut feeling because you actually grow them, and from when you grow them, what do you think? Like, do you see really any big difference from giving a commercial diet, replacing that with something else? Did they all look very normal to you, or is it just that sort of?
No, for now, I will say, like, there should be more measures to be taken, like life cycle assessment should be done for these ingredients, like how much they have, because we are just expecting like they will have this kind of low environmental burdens and low cost effectiveness, but we do not have this kind of studies done on this one. So I think, like, to suggesting to a company, there is a long way to go, because we have to do like economical analysis, and then we have to do like this life cycle analysis. But for now, if I consider like if you use like soybean meal, because we also have a very less options. If you consider the unconventional traditional meals, so they have very less option like soybean meal and then rapeseed oil cake and all those things. So I feel like if we can develop this, then it can be a very cost effective, because we are just using it from the waste. We are just reducing the cost at this point only.
Okay, thank you. Just to simplify the question again, did any of the
Sorry.
did any of the samples die when you did your experiment? They all survived?
Yes, they are, because you see, like for those I obtained from the municipal waste and the municipal waste and the cellulose, they are included in very low inclusion levels.
Yeah, okay, yeah.
So they do not show any kind of adverse effect.
All right, thanks. So we have now plenty of questions here. So the one question from Simi Rose. So the question is, all these ingredients are adding in a very small percentage. Can we suggest these as alternate protein source for fish meal? Is that right? Is that argument in the alliance? Oh, yeah, that's the question.
The thing is, like, I only suggested Neurospora intermedia to be used as a protein source because we did the digestibility study for it. And for rest of the ingredients like yeast and the cello oligosaccharides, so they are used more as an additive in the experiments. Like in my experiments, they have been used as an additives, not as a main ingredient. So only Neurospora intermedia is used as a main ingredient, in which we have seen they are doing good as compared with the control diet in terms of digestibility. So now we can suggest, because when they have a good crude protein or crude lipid digestibility, good level of, then we can take it further for more nutritional study like replacement of or alternative protein.
Yeah, good. So the next question is by Bhaskar. The question is, considering economical point of what is the cost of feed for preparing 1 kilogram feed and fish species recommended like only I guess micus, trout or other cold water fishes. As fungal species in ingredients, is there any prone to fungal disease? Yeah.
First, I will take this question like fungal disease one. So that's why we are doing gut microbiota study and we are looking into the gut health. We are looking into the immune parameters because we know like these ingredients is used for the very first time. No previous studies has been done. We do not have any data. And also it is from a waste. So it should be screened properly. And that's why we looked into a gut hemostasis or gut microbial composition, because whatever there is a problem in the physiology or ecosystem of a host, it should be reflected in the gut or immunological parameters if it is leading to the dysbiosis. I think I'm clear about this question for the fungal. And considering economical point of
Yeah, I think it's all pointing to the same question. Yeah. So I think that question was primarily about, what's the likely that it would get a fungal disease. So I think you nailed it. So the other question from Mukta, I think it's same as what I asked about the economic aspects of the work. Do you want to add any extra comments to that? People are interested to know how the economics work with the. Anyway, I think you answered it partially because your experiment was more on testing these. Oh, sorry. So the testing these as additives and not as a main ingredient. So I think that that put that question a little bit on the off the grid. But if there are any additional comments, Aprajita, you want to make?
It is more like because here I see like, because it maybe my project is little bit like a mix of two kind of like one is like main ingredient and one is like a prebiotic like additive. So people might get confused, and I also get sometimes confused with all these changes. But for me, for now, like I will say it's difficult because I struggled a lot with the ingredients like the supply of the ingredients. So I have to change my research plan as well, because I never get what I was expected to get in terms of microbial biomass for Yareville apolatica and for cellulose-oligosaccharides. So it's I will say it's not very easy now, but maybe in future it can be scaled up or maybe easy. And also like for municipal waste thing and the heavy metal analysis, we have not done heavy metal analysis on this.
So the difficulty of source was just because of your location, or would it be any different if you were in India?
Difficulty, it was like because you get supply for ingredients, okay, but you do not have that. You take municipal waste or organic waste, but you do not have that much big fermenter in the university right now to do the fermentation and to produce that much amount of material which should be needed to running the experiments for a very long time. That's the thing, first of all. And also it was more like sometimes you get, it can be also a future problem, and it is a problem actually. Sometimes you get a batch of organic waste, but you do not know the nutritional profile, because every batch will have a different kind of nutritional profile. So you have to adjust your production of the microbial biomass. Hope you understand, because this microbial biomass goes on the substrate, and the substrate is municipal waste or organic waste kind of thing. But you cannot say every time we will get the Yareville apolatica of the same kind of nutritional profile, or the protein content or lipid content in the Yareville apolatica will differ according to the composition of the substrate. So to maintain this thing for a long term, that could also be a problem.
Yeah. I think we get that. You get your point. Yeah. Sajina have a question. I think it's sort of similar. So her query is how easy and feasible to isolate lipolytica from municipal streams for a large scale use. So I think partially.
So yeah, actually this is like being a nutritionist, I cannot go into the fermentation thing. So there is a different lab who is just doing a fermentation kind of stuff. So they have fermenters and all kind of chemicals. So they bring a specific amount of organic waste from the municipality or somewhere. They keep it in the fermenters and they ferment and they produce. So they store it for some time, or they have it from whenever they need it from that place. So it's like a continuous work to ferment it. So for the first for Neurospora intermedia, I know, like, because we need like 9 kg of the ingredient. So it took that person like because I collaborated with that PhD student, and he did all the fermentation and making Neurospora intermedia, and it took like 3 months for him to get that amount, like 9 kg, and when he was doing alone. So, and not every day, like maybe in batches he was doing. So it took him like 3 months. And for those Yareville applicator, because it was in less amount, it doesn't took longer. So it was very less time.
So Bhaskar is asking, is there any antibiotic residues and heavy metal tested while taking samples for the study?
No, no, I don't think so. It has been done.
No, okay.
Maybe I do not know, because this is my project is a part of a big project. So maybe I don't know exactly.
Yeah. So just one question from my side. So I remember you said you took tissue samples and blood samples, right? So is that blood samples? So most of your work were based on that tissue samples, right? Or I think
Yeah,
that only the oxidative stress is one which I strike me probably from the blood samples. Is that right?
it's from the serum. Yes.
Yeah. Okay. Yeah. We are now saying very nice presentation. Any comparison made with natural environment? Is that clear, that question?
No.
Yeah, so maybe if Naya can just rephrase that question, or even we can unmute and ask the question.
And And also I would like to mention here, because some people are asking about heavy metal or some microbes, like infectious microbes, are there or not. But the thing is, when they collected, like they took a sample for municipal waste, and they fermented it, and they extracted several strains from there, and they did all kind of profiling studies, whatever the studies required for the fermentation research. They did all those kind of studies, but they extracted several species of Yarrow lipolytica, and there was also Klebsiella, some different kind of bacteria also. There were 3-4 bacteria which were isolated, but we selected these only bacteria for the study because of the nutritional profile. So there were also other bacteria which has been isolated, but they were not good for the nutritional study. And then after isolating the strain, these bacteria are grown in the lab in the fermentation using the synthetic media. So it is only the strain which we get from there, and then we grow it in the synthetic media in the lab. So it's not like we are doing everything from the sewage, but we are just extracting the novel bacteria from there.
Yeah, thanks for clarifying that. There are many people who appreciated that it's a good talk and not listen. Giving the names of everyone, but yeah, you can go through that. But just focusing on questions. So Biju Samkavalam have put a question there saying, could you suggest specific microbial biomarkers for rainbow trout gut welfare based on your experience?
For immune gut markers, it could be MUC2, IgT, immunoglobulin T, and there could be like gut integrity genes like occludins, and also there could be like anti-inflammatory and pro-inflammatory genes like IL-10, IL-1 beta, so interferons. So this could also be. So there is a lot of research already done in salmon with a wide range of immune markers, but these are some of them, also in rainbow trout.
All right. Very general question by Ansar Latif. I apologize for joining late. I work on using aquatic macrophytes as an alternative to fish meat. Do you think this field is promising and really sustainable? And added to that, can I please have a copy of this presentation? So in regards to your second question, we will, if Aprajita is happy, we'll be putting this this video in YouTube. We have a YouTube channel. You can find the link from our website, www.afstalks.org. Yeah, over to you, Aprajita, for the first question. Is it sustainable?
For me, when we know something and not the complete chapter, until then we can consider it sustainable, until we get like clear no in future. But still now there is a lot of research to be done to say it's not sustainable. Because for now we are just putting our first step in this direction with this kind of ingredients. So it will be very early to say whether it's sustainable or not.
The other question is by Govind Raju. Highly appreciated about your work. Question is, what's the recommended inclusion rate of intermedia in the commercial diet? I think you haven't investigated in those lines, but you can comment.
What is the inclusion level of Neurospora intermedia?
Yeah, in commercial diet.
Mm-hmm.
Yeah.
It depends like what is the protein content in that Neurospora intermedia strain. So depending on that, we have to decide. Like it should be around like 45. protein. The neurospora intermediate protein content should be at least like 45 to 50%. Then it could be included in the fish diet.
All right.
Replacing fish meal.
So if anyone want to clarify or put the question again, you can unmute and actually put the question across. So Ansar is saying, I don't think this field is new. I chose this PhD topic after doing a literature review of 70 years. I think he meant to say he had gone through papers from last 70 years.
Yeah. Mm-hmm.
Anyway, any
Yeah,
comments?
but I think like earlier, like now I feel like the trend was not very popular that time, but now it's getting more popular with more diversification of the analysis with the life cycle assessment and economic analysis. Because if we do that, then we can say very confirmly like how to proceed with these ingredients. So there are, because the work has been done for 70 years, but it was on a stagnant stage. But now again, this trend is coming back. So it depends on the time.
Are there many people in your lab working on this sort of things?
Yeah, yes. For me, like when I did my PhD, I was only the PhD student in my group.
You're the pioneer then, yeah? Yeah,
Yeah, maybe.
pioneer.
But yeah, now there are more people joined as a PhD.
Yeah. Do anyone else have a question? If you can unmute and ask if you have. There was one of your result was showing that I think it's the second one where you showed the number of lactic acid bacteria increased over the 30-day period.
Yes.
Is there any, well, I don't know much about the topic, but I'm just wondering, is that a good thing or a bad thing, and whether it have any side effects?
I think it's a good thing. Like if the lactic acid bacteria is growing, like we also eat yogurt or something to increase the lactic acid bacteria because it improves the gut health. So this is the similar principle, because when there is more lactic acid bacteria, then they produce metabolites which is needed for the stabilization of the gut. They also produce short chain fatty acids, which has a role in immunomodulation in the gut. So they do a lot good, because they produce a lot of digestive enzymes also. So which
just superficially again, in overall, what was your interest in actually looking in the changes in microbial community, with the diet change?
yeah.
What was the prime interest in looking in that specifically, or rather than looking something else? Is there any reason for that? Like, because it looks like very much focused on the microbial, changes.
Yeah, because yes, that's true. It's more like, because now the research is like that, because the gut microbiota influence a lot of fish physiology. If you consider human also, like it is affecting your mood, your hunger, it's affecting your metabolism. So they play a very big role in any animal, if I say. So why not fish? Like it's also important to understand how the different things are. And also if you think like that, whenever you're having something in your body, it's just going into your stomach, and your stomach is filled with microbes.
Yeah. Yeah.
And uh, they will interact with the microbes in a positive or negative way. So you will understand, like, okay, if I'm giving this new ingredient, and which is from municipal waste, or if it is from agricultural waste, then how it's affecting the gut, first of all, because there only it will be digested, assimilated, and absorbed.
Yeah. Yeah, I was just trying to put your talk in a larger context, why we are doing this. That's all.
So maybe I'm not getting your question.
I think it's not probably relevant for this talk. Actually, it's not science. It's so question is, are there any postdoc opportunities in the field? And also want to know who you are actually. Well, yeah, I'll put that YouTube video in the and then you can see the whole webinar. Yeah, if any other questions before we wind up? Yeah, maybe.
Yeah, thank you very much, Dr. Deepak, and as well as Dr. AC. And it is a great lecture, and really I enjoyed this session. And all the best for your future work. Yeah, actually, it was a nice presentation. I think you should commercialize this kind of thing unless you do these works reach farmers. You should work like, okay, yeah, definitely this is the first step. Finally it should reach to the farmer. Farmer, that beneficiary should be there. Okay, I hope another couple of years you should commercialize what you have presented, and all the to you. And good work, Dr. Deepak. Take care of you.
Yeah. Thanks very much. Yeah, he was my colleague from CIP.
Yeah, but I think that his suggestion is really nice and welcoming.
Yeah, I'm really hopeful. Talks is ultimately going to help,
Yeah, I think this is a very good area. If it is commercialized, it will be really helpful, because we need to replace fish meal with anything which cannot be like a plant-based ingredient anymore. So there is a lot of scale-up studies are going for the insects, like they're producing a factories. You're opening a different production system of factories just to grow insects, and then they would like to use it as a feed, is in the feed as a protein source. So these are the things were there earlier also, but they are now planning to really to make it at a commercial level or scale up to a market level. So there are some people working in this area very hard.
Yeah, actually, I would like to add points regarding insect meal. Actually, although they got commercialized a few black fly larvae, etc., but practically it is very difficult, like, because they are unable to provide even 1%, like that is like supply is very less, and cost of production of I mean this black fly, soldier live, whatever you call that, cost is very high, higher than like fish meal. That's why there's some, I mean, drawbacks are there in utilizing insect meal or whatever the technology is. Yeah, 5% replacement is a great achievement as far as science is concerned. But the thing is, whether we will be able to catch up the scale, means 5% means if you calculate India market itself, it is like almost all 20 lakh tons or something fish meal is required. If you replace that one, if you provide that much, I mean, whatever you have developed, really it is a great thing. If you scale it up, it is a big achievement also.
Yeah, I think your point is really okay. Yeah, it's true. Like they are struggling to scale to make it in enough amount to replace. That's true. But they are also at the same time, they understood like there is a problem with soldier fry. Then they are moving to the another kind of insects also, and they are checking like different kind of insects options, like which can gonna do better economically and environmentally. So, and then they will decide like which one should go further. So it is still in the very basic studies, but people are getting a lot of fundings for this kind of research.
Yeah, correct. Anyway, nice presentation, and thanks for sharing your knowledge for all of us. Thank you very much.
It was my privilege also to be on this platform. Yeah.
Yeah, thanks. Okay. Thanks very much for everyone. for joining the talk. And so, yeah, just stay tuned in our Facebook page, and we also have a LinkedIn page. And I think most of our audience are from our WhatsApp group of AFS Talk. So just join our group, and then we'll post the updates for upcoming talks. And I think Aprajita will give another talk to us maybe after a couple of months later on preparing scholarship applications. I think the students will be more interested in that one. All right.
Okay, sure.
All right. So thanks, everyone. So we'll wrap up here. Cheers.
See you. Bye.